Welcome to The Explainer. Today, we're diving into one of the most important and maybe one of the most invisible technologies in all of modern biology, cell culture. It's really the engine that powers just about everything from vaccine development to cancer research, and we're going to break down exactly how it all works. So think about this for a second. It all starts with a really fundamental challenge. How do you study life at its most basic level? I'm talking about the cell, in a way that's controlled and, ethical. Before you can even think about testing a new drug or developing a vaccine, you need a safe and reliable way to see how it's going to interact with living cells. And for decades, this was a massive, massive hurdle for scientists.
And the solution? Well, it's actually pretty elegant in its simplicity. Cell culture. The whole idea is to take cells out of an organism and create this perfect artificial little home for them right there in the lab. This gives scientists the power to grow just vast quantities of specific cells in a totally controlled setting, which, as you can imagine, just blew the doors open for research in medicine.
Okay, so we get what it is in principle, but why is it so incredibly important? Let's take a look at how this one single lab technique has had this massive, life-saving impact on pretty much all of us. The first huge application is in making vaccines. See, viruses, they're basically tiny hijackers that can't replicate on their own. They absolutely have to get inside a living cell to make copies of themselves. So cell culture provides the perfect factory, a controlled environment packed with host cells that could be used to grow enormous amounts of the virus you need for a vaccine. And the impact of this is, well, it's hard to overstate. We're talking about vaccines for polio, for measles, mumps, and rubella, diseases that used to affect millions of people. All of them were made possible because of this technology. This entire public health revolution was literally built on the foundation of growing viruses in cultured cells.
But it's not just about vaccines. Our bodies also rely on thousands of really complex proteins to function properly, and when we can't produce one of those proteins correctly, we often need a therapeutic replacement. Now, while some simple proteins can be made in bacteria, the most complex ones need all this special folding and modifications that only other animal cells, or what scientists call eukaryotic cells, like our own, can actually do. And that is where animal cell culture becomes a life-saving factory.
And we're talking about some incredibly complex biological machines here. Things like factor VIII for hemophilia, or TPA, which dissolves life-threatening clots during a stroke. And get this, a lot of these are produced in a pretty surprising workhorse, a cell line called CHO, which comes from Chinese hamster ovaries. Yeah, it just turns out these hamster cells are exceptionally good at making complex human proteins and doing it reliably and at a huge scale.
Okay, so we've seen why we do it, but how is it actually done? Well, it turns out there isn't just one single way. Depending on the scientific question you're asking, researchers use a few different approaches, and each one has its own unique strengths.
This table really breaks it down nicely. On one end of the spectrum, you have organ culture. That's where you keep a small piece of an organ alive to preserve its 3D structure. It's perfect for seeing how a drug affects a whole tissue. Then, on the complete other end, you have what we typically think of as cell culture, growing huge numbers of just one specific cell type. That's ideal for mass-producing things like vaccines or proteins. And right there in the middle, you've got primary explant culture, which lets you watch cells as they naturally migrate out from a tissue fragment. Super useful for studying cell behavior.
Now, it's important to remember that none of this works if the cells aren't happy. Keeping cells alive and thriving outside the body is an incredibly delicate dance. You have to perfectly, and I mean perfectly, replicate the environment they're used to inside a living organism.
You should really think of it like running a 5-star hotel, but for cells. Everything has to be just so. The environment must be absolutely sterile. A single bacterium can wipe out an entire culture. They need a gourmet meal, which is this special nutrient medium packed with salts, sugars, and vitamins. Many cells also need a surface to attach to, and the climate, the temperature, the pH, the CO2 levels, all of it has to be controlled with extreme precision.
So once you get them all settled in, the cells follow a pretty predictable growth cycle. First up, there's the lag phase. This is where they're just adapting to their new home, not really dividing yet. Then comes the good stuff, the log phase, which stands for logarithmic. This is the sweet spot. This is where the cells are dividing exponentially, doubling at a rapid, predictable rate. It's the perfect time for experiments. And finally, they hit the plateau phase, where growth just levels off because they've either run out of food or space.
So, how do you know if your little hotel guests are actually healthy? Well, a key metric is something called viability, which is just the percentage of living cells. And there's a simple and pretty brilliant way to check this using a dye called trypan blue.
You see, a healthy living cell has a strong, intact membrane that keeps the dye out. It's like a bouncer at a club. But a dead or dying cell? Its membrane is leaky, so the blue dye gets right in, staining it, which makes it really easy to count.
So far, we've mostly been talking about growing these flat, two-dimensional layers of cells. But the future is all about taking this technology to the next level, recreating the amazing complexity of living tissue right there in the lab dish.
This is where something called organotypic culture comes in, and it is a huge leap forward. Instead of a flat 2D layer of cells on a piece of plastic, this technique lets scientists build complex 3D structures that actually mimic real organs. It preserves the architecture, the cell-to-cell interactions. It means you're not just growing cells anymore, you're building miniature tissues.
And the applications for these mini tissues are just incredible. I mean, you can create much more realistic models for drug testing, you can build complex replicas of tumors to study cancer, keep slices of brain tissue alive to advance neuroscience, and even create models of human skin for dermatology research. This is really where science fiction starts to become science fact.
And all of this brings us to one last really powerful question. If we can take a patient's own cells and grow a miniature version of their tissue or even their specific tumor in a dish, what does that mean for the future of personalized medicine? Just imagine testing dozens of drugs on a tiny replica of your body to find the absolute perfect treatment, all before you ever receive a single dose. That is the incredible power and promise of cell culture.